All right, everybody, welcome back to the second part of a two-part lecture series over general chemistry. Today we're going to hit on three major points. We're going to talk about solution chemistry. What is it? What are the rules of solubility and how do different substances dissolve? We're going to follow that up by talking about blood pH, defining what's meant by acid, base, and salt, and then we're going to end it out by talking about the pH scale, why as allied health professionals we would care about pH, and what buffering systems are and why they're so important. So, as always, I've provided you with a set of learning objectives. Live them, love them, learn them. I've also provided you with a lecture handout that you can print out off Blackboard, and you can download it or type your notes, or you can have it printed out and fill in your notes by hand, organize them in some way so you know exactly what material is going to be on each unit exam, because those handouts kind of form the centerpiece of what you're studying for the course. Now, in order to talk about solution chemistry, a good place to begin is just by talking about what a solution is. So a solution has two components. You have what's called a solute, and a solute is a substance being dissolved. In this case, the solute is this blue powder. Before we put the solute into the solvent, which is the liquid that the solute dissolves in, or the liquid that's doing the dissolving, we refer to this solute as being a solid. So we have solid blue powder. We then take this solute, this solid blue powder, and we put it into our solvent. In this case, our solvent is water. We then mix them together, and the combination of the solute plus the solvent makes a solution. Whenever a solution is made from, when a solution has water as its solvent, we refer to that solution as being an aqueous solution. So we have a lot of different aqueous solutions in biological systems. So you're going to hear me refer to things like blood plasma as an aqueous solution, or interstitial fluid as an aqueous solution, or intracellular fluid as an aqueous solution. And what I'm really saying is that there are solutes dissolved in water, right? And that solution is a water-based solution with a bunch of different solutes dissolved in it. So it's not just one solute. In the case of things like blood plasma or interstitial fluid, it's lots of different solutes dissolved in water. But if water is your solvent, we refer to that as being an aqueous solution. Now, another thing to be familiar with when we talk about solution chemistry is we often measure solution in units of concentration. And no matter which concentration unit we're using, whether we use percent or molarity or osmolarity, concentration always has a similar formula. So the unit of concentration is the same across the board. Concentration is equal to the mass of solute over the volume of solution. That mass could be in grams, it could be in moles, it could be in osmoles, but we have a mass of solute over a volume of solution. So if you don't have very much solute within a volume of solution, here's our solute, here's our solvent, we have a solution with a relatively low concentration. As you add more solute to the solution, the concentration of the solution increases because the mass of the solute comparative to the volume of the solution is changing, right? And in this case, the mass is increasing comparative to the volume. So solutions that have a ton of different solute particles in them, or a high concentration of solutes comparative to solvent, are highly concentrated solutions. So I'm going to ask you questions throughout the course like, what do you think happens to the concentration of blood plasma when you become dehydrated? And you go, okay, so when you're dehydrating, you're losing solvent essentially. So you're losing water, but you're retaining most of your solute. That means the volume's going to decrease, but this is going to stay the same. The ratio is going to change and concentration is going to increase. So when you become dehydrated, typically as a rule of thumb, the concentration of blood plasma increases. So just having an understanding of the unit of concentration is important to make those conceptual predictions that you're going to have to make throughout the course of the semester. Now, when we think about solubility, when I say a substance is soluble in water, or soluble in blood plasma, what I'm really saying is, can that substance actually dissolve and form part of the solution, or does it separate out and form its own distinct layer? The rules of solubility are very simple. Likes dissolve likes. When we talk about like in this case, we're talking about the charge characteristics of the solute and the solvent. So we know that molecules that carry a charge, or individual particles that carry a charge, we refer to them as being polar. So anything that carries a charge by definition is considered a polar molecule, right? So ions, anything that's ionically bound, like all of your salts, sodium chloride, sodium fluoride, potassium chloride, potassium bromide, all your salts, because they're held together by charge, are by definition polar, right? Water is polar, sugars are polar, amino acids are polar. So you take a look at your solute and you go, is that a polar molecule? Meaning, is there a charge, or does that molecule carry a charge? And then you look at your solvent. Water is always polar. Water is always a polar molecule. And you go, well, molecules that carry a charge, or polar molecules, polar solutes, will be able to dissolve in water because water is also polar. That's what we mean by likes dissolve likes. Nonpolar solutes, what happened there? Nonpolar solutes, on the other hand, like fats, oils, have these bonds in which there's no charge, and when you put them into water, they will actually separate out because there's no basis for interaction. So, long story short, the rules of solubility are likes dissolve likes. Polar solutes will dissolve in polar solvents. Nonpolar solutes will dissolve in nonpolar solvents. Now, when you think about solubility, there are different mechanisms of solubility. Our inorganic compounds, our acids, our bases, they dissolve by interacting with water, but the way they interact with water is a little bit different than like sugars and amino acids. So I'm briefly going to talk about this. The way that ionically bound compounds or acids or bases dissolve in water is they undergo a process called dissociation. So when you take solid sodium chloride, or any salt for that matter, but we'll use sodium chloride as our example, and you put it into water, right? You have your solute, you mix it in your solvent, and because sodium chloride is held together ionically, the sodium and the chloride are held together by an ionic bond, it's by definition polar, so salts are all polar. You put it into your water, and the way that it dissolves is the sodium actually dissociates or splits from the chloride to form an independently floating positively charged sodium ion and an independently floating negatively charged chloride ion. So we have our cation and our anion, and when you look at these, the AQ means that these guys are floating around in water. So the molecule has split, and you have these two independently charged entities floating around. The way that that happens is because water is polar, it can actually interact with the positive charge on the sodium, and you see here the water kind of forming the shell around it, or the negative charge on chloride. Here the water is forming the shell, but the hydrogens are oriented toward the chloride, and they form these little things called hydration shells around the solute, and they actually split those things apart. They break that ionic bond between the sodium and the chloride, and now you have independently floating sodium and independently floating chloride. Why do we care about the way that salts dissolve in water, and why do we care about dissociation? Because dissociation reactions form what are called electrolytes. And again, fluid and electrolyte balance is very, very important. So this idea of dissociation, having independently floating charged particles, is really important. Now, not all solutes dissociate when they dissolve. When you have other solutes that we're going to look at in a minute, you don't get dissociation. So let's look at dissociation using this GIF. You put your sodium chloride into a solution, and the water comes and it breaks the ionic bond between the sodium and the chloride, and it essentially strips the sodium and the chloride, or the sodium and the chloride dissociate from one another, and you have independently floating charged particles in your solution that we call electrolytes. So salts, acids, bases, they dissociate in solution. They split to form independently floating charged particles. Molecules that are linked together via covalent bonds, like our organic molecules, don't dissociate. So they're capable of dissolving, but they don't dissociate. And the way that they dissolve is, let's say you throw a cube of sugar into water. Sugar is the solute, water is the solvent. The sugar molecules themselves, which are represented by these little goofy looking guys right here, they don't split. They just separate from one another and they move farther apart from one another, but the individual sugar molecules don't dissociate. So you can look at that a little bit differently over here. In other words, if you take solid glucose, which is what this molecule is here, and you put it into water, it'll dissolve. It'll be part of the solution, and those particles will be floating around in the solution. But the individual atoms that make that molecule up don't split from one another. They remain intact because these are linked together via covalent bonds, and water doesn't break covalent bonds. So the molecules move further apart from one another, but they don't dissociate. And that's really, really important to kind of understand. Now, with that quick introduction into solution chemistry, what we're going to be looking at is an aspect of solution chemistry called pH. And why do we care about pH? Well, what you're looking at is a little kid who's suffering from what's called diabetic ketoacidosis. And we all know what pH is to a degree. We know that it has to do with acid or base or acid-base balance. But we sometimes fail to recognize just how important that acid-base balance is. And if you throw that acid-base balance off, you can have profoundly negative consequences at a clinical level. So when we think about blood pH, pH means power or potential of hydrogen. What we're talking about are freely floating hydrogen ions, but even though they're tiny little ions, when there are tons of them around, they can have profound influences on entire systems. It's so important. So when you think about blood pH is anywhere between 7.35 and 7.45. That's blood pH values. When pH goes down, a solution becomes more acidic. So with respect to human biology, whenever pH drops, the blood pH drops below 7.35, we refer to that as being a condition of acidosis. Now, acidosis presents with a lowered level of consciousness, confusion, weakness, dizziness, and the danger is that you can slip into a coma and die, right? So those would be symptoms of acidosis. Alkalosis, on the other hand, is whenever pH rises above 7.45, and it presents totally differently. Agitation, inability to sleep, anxiety, irritability. So they present completely differently, but the bottom line here is that our pH is within a relatively narrow range, and if it drops, becoming more acidic, or increases, becoming more basic, that can influence everything, and we're going to talk about how that happens. So, what is pH? pH means potential or power of hydrogen. What you are actually measuring when you measure pH is the concentration of free hydrogen ions in solution. So it's the concentration of free hydrogen ions in solution. That's what pH is. It's a measure of the concentration of free hydrogen ions in solution. Ignore this negative log of the concentration of hydronium ions in solution. We aren't in a chemistry class. It's good enough to know that it's the concentration of free hydrogen ions in solution, and what it means is power or potential of hydrogen. So when you think about what influences pH, let's look at how we define our different compounds. Now, here we have a compound called hydrochloric acid. First, I want you to look at the way that that chemical formula is written out. It begins with a capital H, and then it ends with whatever, right? In this case, chloride. This is hydrochloric acid. When you put this hydrochloric acid into a solution, it's going to dissociate, just like that salt did, to form free hydrogen ions and whatever the anion is. In fact, the definition of an acid is any substance or compound that dissociates in solution to form free hydrogen ions. As hydrogen ion concentration increases, pH decreases, making the solution more acidic. Bottom line for this is what you are looking at here is an acid. You know it's an acid because when it enters into a solution, it produces free hydrogen ions. So as you're going through that little handout that you have and you're looking at the problems in follow along 3 that are asking you to complete the dissociation reactions, or follow along 2, I can't remember. If you take a compound that begins with H, right, and let's say it ends with Cl or whatever it is, and you put it into solution, if it dissociates to form free hydrogen, and this would be the subscript AQ to indicate that it's floating around in water, and free chloride with the negative charge subscript AQ to indicate it's floating around in water, any substance that produces free hydrogen ions in solution is an acid. On the flip token, any substance that produces free hydroxide ions in solution is a base. Now that's actually not a good definition of a base, but any substance, like when you're looking, you see potassium hydroxide or sodium hydroxide, if you see this OH linked to metal, not only is that a base, it's a really strong base. Like potassium and sodium hydroxide will melt the skin off your hands if you're Tyler Durden, or are you? I don't know. So you throw potassium hydroxide into solution, it dissociates to form the cation and then the hydroxide anion. This hydroxide anion, what it actually does is when it combines with hydrogen, it forms water. So a better definition of a base, one definition, it's called the Arrhenius definition, is any solute that dissociates to form free hydroxide in solution is a base. But you can also look at a base as anything that removes free hydrogen ions from solution. So the way that hydroxide works is it actually reacts with hydrogen to form a molecule of water, which has a neutral pH. Salts, on the other hand, always consist of a metal and a nonmetal bound together ionically, and when they dissociate in solution, they form a cation and an anion, neither of which is either hydrogen or hydroxide. And in that case, you are looking at a salt. Now, in this dissociation reaction, remember we're putting solid potassium chloride into solution to form aqueous, right? Meaning that these particles are now floating around in a water-based solution independently as charged potassium and charged chloride. We care about all of these because these are electrolytes, and fluid and electrolyte balance is really important. So be able to distinguish an acid, a base, and a salt, and be able to justify why you're calling one an acid, a base, or a salt. Now, when you look at acids and bases, notice the chemical formula of acids. It's not across the board or always true, but they do tend to have this chemical formula in common where they begin with their H. Bases, on the other hand, we see the metal linked to the OH. Now, sodium hydroxide, potassium hydroxide, really strong bases. But a base is just a compound that removes excess hydrogen ions from solution. So a base doesn't necessarily have to have that hydroxide. In fact, anything that removes hydrogen ions from solution is by definition a base. So this down here is a really important base called sodium bicarbonate, also called baking soda. It removes hydrogen from solution. Now, free hydrogen ions can also be called protons. This makes sense if you go back to the periodic table and you think about what a hydrogen ion is. So hydrogen consists of one proton. That's why it's number 1 on the periodic table. By definition, that's its atomic number. And in order to balance that charge, it doesn't need neutrons because you don't have to glue any charged particles together. So you have one electron. When you remove that electron, what you're left with is a proton. So proton floating around, hydrogen ions are just individual or single protons floating around with that positive charge. So when you hear like proton pumps, those are also hydrogen pumps. You can call them either one, just so you kind of are familiar with the terminology. Now, when you look at the pH scale extends from 0 to 14 with 7 in the middle. Now, 7 is what we consider neutral pH. Pure water, right, hypothetically or theoretically has a pH of 7, a pH value of 7. If it's pure 100% water, pH value of 7. Anything below 7 is considered acidic. Anything above 7 is considered alkaline or basic. So you have 7. Anything below 7 is considered acidic. Anything above 7 is considered alkaline or basic. And I want you to understand the relationship between hydrogen ions here, and this is a conceptual question I'm asking you, and pH. So if you put a compound into a solution that dissociates to form free hydrogen ions, the concentration of hydrogen ions will go up. As the concentration of hydrogen ions increases, pH decreases. So they're inversely related. Whatever happens to the concentration of hydrogen ions, the opposite happens to pH. So hydrogen ion concentration goes up, pH goes down. As pH goes down, a substance or a solution becomes more acidic. So hydrogen ion concentration increases, pH decreases, making a solution more acidic. On the flip token, as hydrogen ion concentration decreases, pH increases, making a solution more basic. When you look at the pH scale, you'll notice that we have 7 to 6 to 5 as it becomes more acidic, or 7 to 8 to 9 as it becomes more basic. And we see different substances. I would take a moment to look at these, like human blood pH, 7.35 to 7.45. So our blood pH is slightly alkaline, and it's good. That's where our enzymes and all the different little functional components of our bodies function most optimally. The reason we say that somebody is suffering a condition of acidosis when their pH drops below 7.35 is we are referencing blood as our standard, not the neutral pH of pure water, just so you guys have an understanding of why we call that acidosis and alkalosis. So hydrogen ion concentration, pH, and whether a substance is becoming more alkaline or more acidic is very important. It's also important to understand that the pH scale is logarithmic, meaning it works on bases of 10. In other words, when you say that the pH has shifted from 7 to 6, because it's a logarithmic scale, a solution with a pH of 6 is 10 times more acidic than a solution with a pH of 7. A solution with a pH of 5 would be 100 times more acidic than a solution with a pH of 7. A solution with a pH of 4 would be 1,000 times more acidic than a solution with a pH of 7. It's not a linear scale, it's a logarithmic scale, and we use logarithms to condense widely varied data. But what that's kind of saying, if you have something with a solution of a pH of 6 and a solution with a pH of 7, you're saying that there are 10 hydrogen ions for every hydrogen ion here, right? That's another way to look at it. So it works on logarithms. The small changes in pH reflect large chemical changes, in other words. So somebody's blood could be a pH of 7.2, and you go, well, that's not really that acidic. But remember, this isn't a linear scale. It's a logarithmic scale, and it's changing by bases of 10. So that's actually quite acidic for blood pH. It's important to realize that small changes on the pH scale reflect large changes at the chemical level. Now, ways to measure pH in lab, you're going to see this week's lab is a follow-along lab, and we're going to have a pH probe that we physically stick in our solution, and we're going to get a numerical readout on that pH probe. So when you watch the lab video, you'll have a better idea of what to expect from that. Or depending on the campus, we could use what's called pH paper. And this is a strip, essentially. You dip into a solution and then you allow it to sit for a minute, and you compare the color on the strip to a pH legend. And these are what are called chemical color indicators. They were discovered in flowers, actually. So if you get soils with certain pHs, like really alkaline pH soils, you tend to have like purple and blue flowers, as opposed to when your pH of the soil is a little bit acidic, you tend to have like red flowers or orange or yellow flowers. So it's kind of interesting how pH paper was discovered. So we could use either one of those mechanisms, and it depends on the lab, so watch the lab. This lab, this pH lab that correlates with this lecture, is a follow-along lab. So you're actually going to be taking down data points and answering questions on a formal lab report, which, trust me when I say, become important when it becomes exam time. Now, follow along 5, why is pH so important biologically? So pH, which is the power of hydrogen, represents the concentration of hydrogen ions in solution. Why do we care about it? Well, pH profoundly impacts the solubility of certain substances. It influences whether substances will dissolve or not in solution. pH affects the structure and function of proteins, notably enzymes, and you're going to find that proteins are a really important class of biological molecule. They're like the little molecular workers of the cell, and they have a wide array of functions. We are protein-based organisms. And all organisms on this planet can only survive within narrow pH limits. So every organism exists within an ecological niche. Our pH niche is between 7.35 and 7.45, and we work to regulate our pH. Our external environment, the pH can't vary too wildly, or it'll influence our internal pH. So organisms are adapted to environments with certain pHs. Now, when I say solubility, let's take a look at the enamel in your teeth. So, any solution with a pH above 5.5 is not going to impact the enamel on your teeth. So you can drink tap water, which a lot of times is a little bit acidic depending on the aquifer you get it from. You can drink certain juices, etc., without having any kind of negative impact on your dentition, with the exception of the sugars, and that's another discussion altogether. But once you hit this point of what's called critical pH, all of these solutions with a pH below 5.5 or more acidic than 5.5, once you hit 5.5, the enamel in your teeth becomes soluble, right? So it changes the solubility characteristics, and it actually starts dissolving out the enamel in your teeth. This is called diet soda mouth. Right? It's also called meth mouth. Look at the similarities there. So if you're drinking acidic beverage all day, what you're doing is you're wearing away the enamel on your teeth. And that's what I mean by solubility can change. We care about pH because it influences solubility, right? So you can start getting things like body structures physically wearing away in response to pH stress. Or bone. Say you eat a diet that's too high in protein, or you have a metabolic disturbance that drops the pH of your blood. Well, when the pH of your blood and the fluids in your body drops, it can actually start physically dissolving away the components that make up bone. And when those components that make up bone start dissolving away, you can start treading into disease states like osteoporosis, which is just a condition of porous bone because, in some cases that bone just physically dissolves away. That's how lifestyle factors can be linked to diseases like this, especially in men. So we care about it. We care about solubility because you're talking about the stuff that physically makes you up. And if you shift pH, you can start dissolving that stuff out. And that is no good. We care about pH, number 2 reason, because it influences the function of proteins. Now, we haven't had our unit discussion on proteins yet, but in general, proteins are little pieces of molecular machinery. So they're little molecular machines that do stuff. And if this was an enzyme, it is an enzyme, right? Enzymes are like little biological scissors or glue guns. They can break stuff down or build stuff up, right? They catalyze chemical reactions. Now, if you remember from the first discussion, I was talking about if I was an enzyme and I was standing next to a cell, the cell would be like 7 Empire or 10 Empire State Buildings tall and 10 Empire State Buildings wide, and there'd be millions of me in there. So enzymes, proteins, much smaller than cells. But they're the workers of the cell. They're little molecular workers. And the 3-dimensional shape, the structure of an enzyme is really, really important to what it actually does. So this enzyme is responsible for breaking maltose down into glucose that can be absorbed along the GI tract. The only way it can do that is by binding to this maltose molecule, which fits into a little part of the enzyme called the active site. It stresses those covalent bonds and it splits that maltose into its building blocks, which are 2 glucose molecules, which can then be absorbed along the GI tract. Now, it's key that this enzyme maintain its shape in order to carry out its function. When pH changes, when pH either becomes more acidic or more basic, what it does is the concentration of hydrogen, those free-floating hydrogen ions, can influence the shape of proteins. In other words, it can cause their shape to change, can cause them to unravel or essentially to lose their shape. So when a protein loses its shape, we call that denaturing. And you'll notice that here we have the rate of reaction. So how quickly does this particular enzyme or whatever enzyme we're talking about, how quickly does it do its job? How quickly can this break maltose down? When pH is ideal for whatever enzyme you're talking about, those enzymes work really, really well. They work at a high rate, they're cranking out, they're cutting stuff down or building it up. But if you throw pH off, you change the shape of these proteins, the rate of the reaction can go down to 0. And that's because these proteins are denaturing or unfolding. They're losing their shape. So when you think about proteins, we tend to abstract them, but here we're looking at activity inside a cell. And we're looking at what's called a motor protein. And the job of motor proteins is to drag sacks of material around in the cell. This motor protein is physically like a little worker. It moves. It has these little legs. I mean, it really looks like this. It has these little legs that physically move back and forth, and that's how it can drag this little sack of stuff called a vesicle within the cell. So this is happening at the subcellular level. We're talking about really small things here. Notice its shape. Its shape is important to what it does. These other enzymes over here, some of them are building stuff up, some of them are breaking stuff down, and you have millions of these little proteins in every cell, and you have trillions of cells, and these proteins are just going around doing all of this important stuff. If you throw pH off, you're going to change the shape of the protein and you're going to ruin its function. So when a protein is unfolded, or you cause it to unravel, or you change its shape, we say that protein is denatured. There are a couple of things that can denature proteins. Heat can denature proteins because you break the hydrogen bonds that hold the proteins together. And pH, right, shifting the concentration of hydrogen ions in a solution, right, can break the bonds that hold proteins together, causing them to denature. Now, to de-abstract the process of denaturing of a protein, let's think about cooking an egg. So you crack an egg and you put this egg on the skillet, and initially the white of the egg and the yolk have this kind of gelatinous fluid-like matrix, right? And it's typically clear, so this kind of clear fluid in the white of the egg. As you cook the egg, what you're doing is you're taking these globular proteins that were floating around in the white, the egg white, and in the egg yolk, and you're unraveling them. You're breaking the bonds that hold them together using heat. And when you break the bonds that hold those proteins together using heat, you essentially ruin the shape of the protein or denature the protein. And denatured proteins have this tendency to stick together, right? Because they're no longer little independently functioning machines. As they stick together, the consistency of the tissue changes. So when you're cooking food, what you're really doing when you cook food is you're denaturing proteins in that food. This guy down here had acid thrown on his face, and what happened is this change in pH, those hydrogen ions caused these proteins to break apart, coagulate, change the texture. Essentially, they denatured proteins. In other words, when you talk about shifts in pH from a clinical perspective, let's go back to this kid up here. You have this kid who you run the electrolyte balance on him and you run the pH values and you go, oh, the pH is like 7.1. That's an extreme acidosis because remember that pH scale changes a lot. It's a logarithmic scale, meaning bases of 10. And you're looking at him and he's in bad shape. What's essentially happening when pH drops and you enter into a condition of acidosis is the proteins in your body are denaturing. In other words, you are cooking alive, and you want to correct that as quickly as you possibly can, especially if the disturbance is major, because you don't want your patient cooking alive in front of you. That's what we're talking about when somebody's in a condition of acidosis. Their proteins are denaturing, and if too many of them denature, they're not going to be able to recalibrate and regroup, and that's massively problematic. So we care about pH because it influences our little molecular workers, proteins, and it can change their shape. And proteins have an ideal pH in which they exist, and if you throw that off, it becomes problematic. if you've ever cooked food using pH, like you can cook fish using lime to make ceviche, right? You get that change in the consistency or the texture of your food. When you cook food, what you're really doing is you're denaturing proteins. Finally, organisms exist within ecological niches. And these ecological niches, one of the key defining points of them is their pH. So, you know, you have little marine organisms that exist, in a slightly alkaline pH in some of their marine habitats. One of the problems is whenever carbon dioxide is emitted, carbon dioxide can dissolve in water. And when it dissolves in water, it reacts with water to form carbonic acid, which dissociates to produce free hydrogen ions. In other words, the more carbon dioxide there is in a system because of this particular reaction it has with water, the more free hydrogen ions, meaning that that solution, as hydrogen ion concentration goes up, pH goes down, making the solution more acidic. So when you're burning fossil fuels all the time and there's not an appropriate cycle to kind of take up that excess carbon dioxide, it dissolves in water, like our ocean water, reacts with water to form carbonic acid, which dissociates to produce free hydrogen. It drops the pH of, let's say, oceans or lakes, and it throws that pH off. Well, unfortunately, marine mammals and little crustaceans and organisms that live in reefs, right, these little plankton and things like that that live in reefs rely on relatively stable pH values. So when those pH values shift, those organisms die, and what you're looking at here is essentially a bleached reef. And that's why ocean and lake acidification is such a concern, because you worry about all those organisms that have evolved or live in these relatively stable pH environments. When you start throwing that pH off, you're going to have really profound impacts on the ecosystems. So follow along 6 starts by asking you, well, why do we have carbon dioxide in our body in the first place? Well, let's look at what happens here. So plants take carbon dioxide and water, which are inorganic molecules, which we'll talk about later, and energy from the sun, and they use that energy from the sun essentially to build glucose, with the byproduct being oxygen. So all glucose and oxygen, meaning the things we need to live, come from plants. They come from a process called photosynthesis. Then in our cells, we take this glucose and oxygen and we burn glucose in the presence of oxygen in order to make energy in the form of ATP. So we burn glucose in the presence of oxygen in order to make ATP in the process of cell respiration. The byproducts of burning glucose in the presence of oxygen, just like burning gasoline, are carbon dioxide and water. Now, carbon dioxide reacts with water in biological systems, meaning in aqueous solutions, so in things like your blood plasma, to form carbonic acid, which dissociates to form bicarbonate and free hydrogen ions. And we care about this. So where does that carbon dioxide come from in the first place? Well, when we burn sugar in the presence of oxygen in order to make energy in the form of ATP, carbon dioxide is the byproduct of that. And we call that process in which we burn sugar in the presence of oxygen in order to make energy cellular respiration. So carbon dioxide is the byproduct of energy production in your cells. So we inhale in order to get oxygen, we eat in order to get glucose, and then we burn that glucose in the presence of oxygen to make energy. And the byproducts of that energy production are carbon dioxide and water. So we need to get rid of that carbon dioxide. Now, in order to prevent dramatic changes in our blood pH, we need what are called buffering systems. Buffering systems are solutions that resist change to pH. Buffering systems always consist of 2 parts. You always have what's called a weak acid and a weak base. So buffering solutions are solutions that resist changes to pH. Being able to resist changes to pH is really important, especially in biological systems, because most things in biological systems are pH sensitive. They work best within a narrow range of pH limits. So It's always, all buffering systems consist of a weak acid and a weak base. We'll talk about what constitutes a weak acid and a weak base in just a moment. And notice that this reaction can go in either direction. And the long story short with buffering systems is if you add too much hydrogen to your solution. So let's say you drink something like a Mountain Dew and you start adding hydrogen, free hydrogen to the solution. What this buffering system will do is that base will activate and it will take that free hydrogen and it will store it in the intact weak acid. In other words, with buffering systems, if the pH drops too low, meaning you add hydrogen, right, the weak base will activate, removing that hydrogen from solution. On the flip token, if you remove too much hydrogen from solution and the solution becomes too alkaline, this acid will activate and it will add free hydrogen to the solution. So when the base activates in this system, you say that the buffering system, the equation will shift to the left because you're getting rid of the free hydrogen as that chemical system shifts to the left. If you wanted to add hydrogen, let's say like you're taking too many Tums and your pH gets thrown off and your buffering systems kick in, that weak acid will activate and it will dissociate, adding hydrogen to the system. So that particular chemical reaction will shift to the right. Now, really, really useful. Buffering systems are incredibly important, and it's because the proteins in our body and the different structures that make up our body function best within a narrow range of pHs. So when you think about how buffers work, you always have a weak acid and a weak base. The reaction, because of that, can always go in either direction depending on the stress to the chemical system. So this is called an acetate buffer. You don't need to know that, but the bottom line is if you add hydrogen ions to the system, meaning you add something acidic, that system will shift to the left in order to get rid of the hydrogen ions. In other words, the weak base will activate. If you take away hydrogen ions and the solution becomes too alkaline, this system will shift to the right, adding free hydrogen ions in order to keep that pH within a narrow range of limits. In other words, you can activate the weak base or the weak acid. Acids or acidic insults will activate the weak base. Basic or alkaline insults will activate the weak acid. So let's look at this most important buffering system in our body, the carbonic acid bicarbonate buffering system. This carbon dioxide comes, it's a byproduct of burning glucose for energy. So just like when cars burn gasoline, they emit carbon dioxide, we also produce carbon dioxide in our cells when we burn glucose for energy. When carbon dioxide reacts with water, it forms carbonic acid. Carbonic acid dissociates to form free hydrogen and bicarbonate. This buffering system that consists of this weak acid, the carbonic acid, and this weak base is called the carbonic acid bicarbonate buffering system. Sometimes textbooks will shorten it just to show you this. And how does it work? Well, focus on this side of the equation. Just focus on this for a minute. If the solution, let's say your blood plasma becomes too acidic, it means that hydrogen ion concentration has increased, right? That's the definition of acidic. So hydrogen ion concentration increases, pH decreases, solution becomes more acidic. When hydrogen ion concentration increases, you activate the weak base and the reaction shifts to the left, right? In other words, that base reacts with that hydrogen, pulling it out of solution. When the solution becomes too alkaline, meaning hydrogen ion concentration decreases, pH increases, and it becomes more alkaline, let's say, than 7.45 blood pH, then this carbonic acid activates, it dissociates to form the free hydrogen and the bicarbonate ion, and you add those free hydrogen ions back. In other words, the system shifts to the right. And buffering systems are just a back and forth shift. They're just shifting back and forth all the time. Now, Why do we care about it? Again, look at like Mountain Dew, aka just poison. It's just poison is what it is. I'm not sure why anybody drinks it. But it has a really, really lower acidic pH, and it can erode the enamel in your teeth. So can any of these. I just highlighted Mountain Dew because it's my least favorite of beverages. So, now, we've talked about buffering systems. Buffering systems are systems that resist changes to pH. They always consist of weak acids and weak bases. In response to an acidic insult, the base will activate, shifting the reaction to the left. In response to a basic insult, the acid will activate, shifting the reaction to the right. What's the difference between a strong acid and a weak acid? So if we look at this, let's pretend that the blue represents the hydrogen ion, and the red represents whatever the anion is. It could be anything. I mean, there are literally thousands upon thousands of acids. When you throw an acid into a solution and all of the hydrogen ions that are available dissociate, meaning you split to form positive and negative, and 100% of those molecules split, we say that that particular acid has a 100% dissociation. Strong acids are acids in which each individual molecule, that hydrogen ion, will pop off and enter into solution. That's what's called a strong acid. It has 100% dissociation. On the other hand, a weak acid like carbonic acid, let's say we throw carbonic acid in and we have the hydrogen that should dissociate. Well, with weak acids, most of the molecule remains intact, right? And only a couple of the molecules actually dissociate. And because of that, because it has a low percentage of dissociation, you're not going to add as much free hydrogen to the solution. So we refer to those as being weak acids. It refers to the percent dissociation, how many of those molecules split. Now, the final topic for today is neutralization reactions. Neutralization reactions happen whenever you take a strong acid and react it with a strong base. So if you take a strong acid and react it with a strong base, the hydrogen will pop off of the acid, the hydroxide will pop off of the base, and the hydrogen and the hydroxide will react to produce water, H2O. Then what you have left is the cation and the anion, in other words, the metal and the nonmetal, and a metal and a nonmetal together, right, compose a salt. So when strong acids react with strong bases, they neutralize one another, and the product of a neutralization reaction will always be water and salt. So when you're completing the reactions that I've given you, think about that. Think about those different things we've talked about. Thank you very much for your time. Have a good day.